Welcome, everyone. Thank you for joining us. We have Dr. Jenifer Walke with us tonight. Um, Jenifer is an associate professor of biology at Eastern Washington University. She's a microbial and disease ecologist studying the roles of beneficial and disease-causing microorganisms on their hosts, including honeybees. She earned her Bachelor of Science degree from James Madison University, a PhD from Virginia Tech, and was a postdoctoral researcher at Virginia Tech in the entomological and biology sciences departments. Jenny began conducting research on the honeybee gut microbiome in 2015 and just wrapped up a National Science Foundation research project investigating the complex interactions between honeybees, their hosts, the gut microbiomes, and the pathogens and pesticides, which we'll talk a lot about tonight. Jenny is the advisor for EWU's Busy Bees Student Club that manages the hives on one of the student center's green roofs, along with the West Plains Beekeepers Association. In addition to teaching and research, Jenny enjoys engaging in community outreach, including with elementary and high school students, and playing outdoors with her husband, 2 daughters, and 2 dogs. You can learn more about Jenny's research at walkelab, WALKELAB.com. Tonight, Jenny is going to talk to us about the role of honeybee gut microbiome on bee and colony health, including the environmental factors that may influence the bee gut microbiome, such as pesticides, disease, and nutrition, and tell us why it's so important and give us some background along the way. So that we're going to turn it over to Jenny and look forward to hear more about bee gut microbiome.
Excellent. Thank you, Courtney, for that introduction, and thank you, WASBA, for the invitation to speak tonight. I'm really excited to be here. I am going to get my slides pulled up here. Okay, should be good. All right. Well, yeah, so I'm going to talk about the honeybee gut microbiome. And to start us off, I have this image here of an auger art work, a masterpiece. So instead of using canvas and paintbrushes, auger art uses petri dishes and agar media and inoculating loops and colorful microorganisms to make art. And of course, this one combines 2 of my favorite things, bees and microbes. So that'll kick us off. And like many of you all out there, we just recently got a couple packages of bees installed on our hives that are on our student center's green roof, and in collaboration with West Plains Beekeepers Association here near Cheney, Washington. And so that was really exciting event, and we have lots of energetic and enthusiastic students to help with that. We're also looking to get some hives on our prairie restoration project, where we're converting kind of old wheat fields that are no longer being farmed into native Palouse prairie habitat. And that should be a great project for pollinators. So we're excited about that going on campus at Eastern. So today I'm going to be sharing a little bit about the invisible world that exists inside of bees specifically. So this is a simplified tree of life showing the 3 domains of life, bacteria, archaea, and eukarya. And as you can see here, the very tiny tip of the tree of life is what we can actually see. So plants, animals, the bees are on that part there. And the rest of living things are invisible or microscopic. But even though they're small, they're mighty and do really important things for their hosts. And another thing to keep in mind with microorganisms is that, of course, we know there are some pathogens that are, disease-causing and are harmful to their hosts. And then, of course, we know there are mutualists that provide some benefit to their host, and then the host, in return, has some benefit for the microbe as well. But the reality is that a lot of microbes kind of exist along this continuum. And depending on the environmental context, they can change depending on their abundance or the temperature or the presence of other organisms. And so that's kind of the one of the big goals of my research lab is trying to understand one, kind of the relationship that microorganisms have with their hosts, including bees, and then also kind of how the environment can influence that relationship. Um, and so the honeybee gut microbiome is relatively simple, although today I'll share some results that we have that indicate that it might be more complex than we think. But this review article and this figure here shows kind of about 8 to 10, up to even 12 bacterial species that live in the bee gut. And they occupy habitat in the different regions of the gut. So we have the crop, the midgut, and then the hindgut, and each of these regions you can see kind of are host to different microorganisms. And we're starting to really understand kind of how they're interacting with the cells of the gut lining and that chemical signaling that's happening between the host and these microorganisms. So it's a relatively simple microbiome compared to the human gut microbiome, for example. And it's also pretty quick to establish, which makes it a good model system too to study, with the human gut microbiome too. And here is a figure that's showing some of the main functions of the honeybee gut microbiome. So there's research suggesting that it's important for the ability of the insect to kind of get nutrients and energy and break down food from the food source. Also influencing their behavior, toxin degradation, which I'll kind of talk about a little bit in terms of the ability of some of these bacteria to break down pesticides that the bees are exposed to. Also, these microbes play a role in defense against pathogens and parasites that I'll also talk about today. And so here's a kind of outline of the talk and some of the latest projects. I'll basically be telling you all 3 stories that that we have been working on in my research lab that kind of are looking at 3 angles of the environment and how that influences the microbiome, and vice versa, how the microbiome can influence, the impacts of these environmental factors on the bees. And so we'll be talking about pesticide and antibiotic exposure and disease and nutrition and kind of diet as well, and how that relates to the structure of the microbiome, which is sort of like who's there, what types of bacteria are there, and how much of each microbe, and then the function, which is what those microbes are doing. So bees, of course, they encounter lots of lots of things in the environment, whether it's out when they're foraging or getting water, or even in the hive and thinking about miticides and these other chemicals and antibiotics that they're exposed to kind of from beekeepers in the hive. And so we're really interested in understanding, do these things impact the microbiome of the bee, and then, does that translate to health of the colonies as well? So starting off, we'll talk about a couple studies that we have been doing with pesticides and antibiotic exposure. Okay, first off, I want to tell you about a survey that I'm really excited to share results that we they're not published yet, but we're hoping to get that done pretty soon in collaboration with my colleagues at Virginia Tech. And with this survey, you can see kind of we have a range of Washington samples and then kind of a transect along to the East Coast in Virginia, in far eastern Virginia. And so with this study, we were really interested in identifying antibiotic resistance genes in the gut microbiome and if that varied across the country. And so, as you may know, antibiotic resistance in general and with humans, it's a huge concern, societal concern with the overuse and the misuse of antibiotics, both for, human healthcare, but also in agriculture and livestock. And basically the bacteria are evolving resistance to traditionally used antibiotics, which then makes it difficult to treat these bacteria that especially are pathogenic. And so it's a big concern. And so we were interested in exploring this in the honeybee gut microbiome. And so what we found is that the antibiotic resistance genes, which we have abbreviated as ARG, the frequency and the composition of these antibiotic resistance genes did vary across the United States. Furthermore, tetracycline was the most commonly found antibiotic class that we found resistance to. And if you know much about the use of antibiotics in beekeeping, tetracycline is commonly used. And so we have identified that in the honeybee gut microbiome, those bacteria are evolving resistance and have resistance genes to this class of antibiotics that's commonly used in beekeeping. So that could become an issue with the use of tetracycline if it's no longer going to be effective against its target bacteria. And so you can see kind of across all of our sampling sites that that tetracycline was the most commonly identified antibiotic resistance gene class. And furthermore, we were able to identify which bacteria in the gut microbiome of the bees had these antibiotic resistance genes. And so that's what this figure is here, is showing kind of the genus of bacteria, kind of those main groups of bacteria, and then whether or not they, the maroon part, I realize I don't have the key on here on this particular figure, but the maroon part is showing the proportion of kind of all the genes in those microbes that are antibiotic resistance genes. And what I especially want to point out is that over here on the far right, those two groups, Gilliamella and Freshella, are bacteria in the bee gut that aren't very abundant. So they're considered core members of the community, but they're just low abundance. And those taxa have, higher proportion of antibiotic resistance genes. So it's not just the really abundant bacteria that have a lot of these genes, it's even some of the less abundant ones too. So that has implications in terms of, quantifying the microbiome structure and thinking about the presence, but also the abundance and whether or not they have genes that are likely important in their communities. So I'm really excited to share these results with you all, and hopefully we'll get them published soon. And then a little closer to home, I want to share a study that we did here in Eastern Washington, and here we were interested in pesticide exposure and whether or not that impacts the gut microbiome. And there's a lot of overlap in the genes that are responsible for antibiotic resistance that I just talked about, and that are also responsible for pesticide resistance due to the mechanisms of how they work at the cellular level. And so what we do when we collect the samples from the hive, and then we do a dissection by just pulling the stinger out, and then the whole GI tract comes out, and then we're able to use that for our sample to do DNA sequencing to look at all of the microbes in the bee gut sample. And so for this study, we sampled 6 locations across the Spokane, Cheney area in association with West Plains Beekeepers. So really grateful for them for this project. And so the first step was to quantify the pesticides and see, are they there and how many hives have them? And so we focused on these 6 pesticides. Some of them are in-hive miticides, like talfluvalinate, for example, that you might have heard about. But imidacloprid, which is a neonicotinoid insecticide, um, and that there's debate and conflicting research out there as far as the impacts on the bees themselves. Um, and of course, that's going to depend on the dose and the environmental context. But it does appear to disrupt their foraging behavior and can lead to death depending on the context. And so because imidacloprid was kind of the most prevalent of the pesticides that we tested, we decided to focus on that for further analyses looking at the role of this on the microbiome structure. And just a note here in the corner, you can see that the imidacloprid concentration that we detected in our hives was comparable to the level that another study found in Virginia to be at that level as well. So that was kind of interesting that, the exposure across the country appears to be, consistent levels at least. So I just wanted to point that out as well. Okay, so this figure, let me walk you through it a little bit. It's basically a way to look at the microbiome of different hive samples. So first of all, each point on the figure represents the microbiome collectively of a hive, where we've sampled multiple bees from that hive and characterized its microbiome. And so that's what each point individually, it's like the microbiome of a bee. And so the closer 2 points are together, the more similar their microbiomes are than points are further apart. So that's one thing to keep in mind. And then also the points, as you can see, are color coded and size scaled based on their imidacloprid concentrations in the bees from those same hives. And then we did a statistical analysis, and that identified a correlation between the levels of pesticide and the microbiome composition as well. And so, and this was in the field, just kind of going out, not manipulating any variables, just going out, collecting, quantifying pesticides in the microbiome, and we're seeing this correlation with the imidacloprid. And we did not see this correlation with the other pesticides that were detected in these samples. So it was just the imidacloprid that we found that. And another way of looking at these relationships is looking at the evenness of the community. So that is a measure of kind of the relative abundance of each taxa, each type of bacteria in the bee gut. And so we found that, with increasing imidacloprid concentration, that we saw decreased evenness, which is a measure of diversity. So the community as a whole became less even or less diverse. And then we also, ran a similar analysis, but instead of looking at imidacloprid concentration as a continuous variable, we just said imidacloprid present, yes or no, and categorically, and that also detected kind of that higher evenness if they did not have imidacloprid. So the imidacloprid appears to be, decreasing the diversity of the communities in the bee gut. And specifically, we identified this taxa called Bartonella apis. It's one of those about 10 bacterial groups in the bee gut that was really driving that difference in community composition. So the bees with imidacloprid had significantly more of this Bartonella apis species. And this is interesting because Well, I guess like it leads to an interesting hypothesis is that maybe Bartonella is able to metabolize or use imidacloprid. And so imidacloprid's present, this species might be able to use it and thus, grow more and an increase in abundance in the community. And this has been found in a couple of other bacterial species, including lactobacillus, that can metabolize imidacloprid as well. And so there are other studies that have shown that these bacteria can actually use and break down these pesticides, and our results are kind of in line with that, which is pretty interesting. And so with that, that was, the field results with not manipulating anything. And we really wanted to understand in more detail what was going on. And so we went into the lab and cultured some of these bacteria and then tested them in lab assays against imidacloprid. So down here on the bottom on the x-axis, you can see kind of increasing imidacloprid concentration. And then on the y-axis is the change in growth of the bacteria. So as you can see, with more imidacloprid, the bacteria did decrease in their growth overall, which isn't too surprising. But what was surprising is the variation that we saw among all these different strains that we had isolated. Um, and so we had, multiple strains of each of these three bacterial species from bees. And yeah, so you can see kind of the level of variability in the impact of this pesticide. And so then the next step is to try to figure out, well, why are these different strains of the same species behaving different in response to the pesticide? And so for that, we have started to do whole genome sequencing to look at the DNA of the whole genome of these different bacterial strains. And this is a little messy, but essentially with, these three different bacterial species, we compared the genome across different strains. And essentially what you want to look at are these different lines that are crisscrossing between the different genome sequences, and that's indicating differences in the genome. And so we have these differences in the genome that are aligning with differences in the response to pesticide, even though they're the same species. And so that's what I was kind of wanted to come back to, that complexity. It's like, yeah, there's only, 10 or so bacterial species in the bee gut, but they do have strain level variation in their genomes and in how they're responding to pesticides, which is really interesting. And further, we're really trying to identify, okay, what specifically on the genomes are explaining that variation. And so we've started to identify plasmids, which are DNA molecules, but they can be transferred across different bacteria. And so we a lot of antibiotic resistance genes and pesticide resistance genes are located on plasmids that can be exchanged. And so that is pretty interesting. And we're also kind of coming back to imidacloprid specifically, this neonicotinoid insecticide. The breakdown of it, the kind of the intermediates and identifying which enzymes are responsible for breaking it down has been pretty worked out before, actually. And so fortunately, we have good targets to then look at our bacterial DNA sequences and try to figure out, okay, what part of the genomes are breaking down imidacloprid, and then how does that vary across our strains. So it's a whole new area of our lab research and doing the whole genome sequencing. And so then if we can find it, then we might be able to ultimately be able to adjust it in a way that makes the bees less vulnerable to the pesticides. And another interesting thing that we found with this study is not only do different strains behave differently, but the overall community structure of the hive microbiome varies across sites and particularly even in very close proximity. So this was a little bit surprising. So remember, this is one of those figures where each point is the hive microbiome, the bee gut microbiome of the whole hive. And so the closer together they are, the more similar they are. And these are samples from that study where they're color coded, right, by the site. And Eastern Washington University is in Cheney. And so we had a site on campus, and then we also had a site in town about 2 miles away from the campus hives. And as you can see here, their microbiomes are different. They're clustering differently from each other. They're not overlapping like some of the other sites that you see. And so your neighbor, who is just a couple miles down the road and they have hives, they have probably the same types of bacteria, but just different amounts of each type, which means that the whole community structure is different, even though again they're able to overlap in their range and so forth. So this was pretty interesting to think about. So different hives can be responding differently to these environmental factors, even if they're in close proximity to each other. So that was pretty interesting. Okay, so that wraps up kind of part one of the story focused on pesticides and antibiotics. And now I want to talk a little bit about disease and specifically a study that we're doing, again, collaboration with Virginia Tech, looking at nosema and microbiome interactions. Okay, so here's a little cartoon diagram, and you all might be pretty familiar with this pathogen that's a microsporidian pathogen, so it's kind of like a fungal-like organism, and it reproduces inside of the gut lining cells. And so it has these polar filaments that allow it to attach to the gut lining and then enter in to reproduce. And then the spores can exit those cells as well. And then of course, we have our bacteria that are certain types that are specifically lining the midgut as well. And so this study was really focused on trying to identify what composition of those bacteria are there, and then how they're impacting the reproduction of nosema. So we've done a series of experiments where we're exposing some bees in a lab setting, and I'll show you some fun pictures in a second. And then we're able to assess kind of susceptible or resistant microbiome structures, and then recreating those protective microbiomes, and then testing whether or not, again, with another round of exposure, if they're protective still. And so we've done a series of experiments. I'll just show a little bit of that work. And there's some interesting pictures of kind of how we've gone about that. But essentially, we've looked at known age bees. So we have standardized the age of the bee in these experiments, and then dose them by, feeding them sugar water that has the nosema in it. And that's how we've inoculated them with a known concentration that we've used a hemocytometer to count the cells that we're exposing them to with a pipetter. And then we've quantified nosema infection intensity and then looked at the metagenome which is basically the all of the DNA in the gut sample, including the host DNA and the nosema DNA and the bacterial community DNA. So just to walk through some of the pictures. So we basically take a frame that has a brood frame, and put it in and get rid of all of the bees, the adult emerged bees. So we just have the brood frame and then put it in a mesh enclosure in an incubator overnight. And then the next day we have, the newly emerged bees. So we know their birthdays. And then we anesthetize them with CO2, and then paint them and mark them, so we can track them when we put them back in the hive. And the reason why we need to put them back in the hive at this point and not just go straight to our experiment is because we need them to get their normal microbiome. And the only way that they can do that is by going back into the hive, because they need that trophallaxis to exchange genetic or exchange the microbial contents, and get their normal microbiome established by 7 days post-emergence. So we had to put them back in to get their normal microbiome established. And then we can go back, 7 days later after that's been established, and find the marked bees and recollect them from the hive, which is pretty fun. And then this is what I was showing with the dosage and how we actually give them the nosema exposure. We can put them in individual little tubes and pipette them in, pipette the inoculum into the bee, and have them eat that, and then put them in these cages, where we can monitor them and have, standard light cycles and humidity cycles. And you can see the little tube here with food for them. And then we can, proceed with the experiment and quantify the nosema levels several days later. And so with this experiment, we were able to successfully increase their exposure level. It's tricky because they already kind of have a baseline amount of nosema. And so it's hard to get kind of a no nosema group, but we were able to get a, higher infection intensity significantly, which you can see with these like darker blue are the nosema ones. And we did this across 3 different hives as well. So we were able to successfully do our nosema infection experiment. And then we were interested in, after we have our different groups, so we have control, low, and high nosema levels, then we could look at the gut microbiome. And specifically with this, we were able to look at different properties or qualities of the microbiome. And I just want to point out this heat map with this bottom row here, which is showing that with more and more and higher levels of nosema, we were getting more of this type 6 secretion system. And so what that is in bacteria, it's basically like the syringe that some bacteria have. It's the structure that they can inject toxins into other cells and affect them in that way. And so it's the structure on their membrane that they can use to inject these virulence factors. And so we found that, so not only are your bees that, or have really high nosema infection levels, and having that to deal with, they're also getting more bacteria that are able to inject these toxins and virulence factors into potentially the host bee cell. And so it's sort of like a double whammy, and kind of a secondary negative effect when bees are highly infected with nosema. So there's a lot going on. And then also this is showing the different types of bacteria and how much of each type they had. And I just want to highlight this green bar, part of the bar here, that we found that bees that had higher nosema levels had significantly higher Freshella in particular. And so kind of our next step is to see, okay, is it that taxa that tends to have more of this type 6 secretion system property? And so the nosema is, affecting the amount of bacteria and these other properties that are probably not good for the bees or their other microbes in their communities. Okay, so lastly, to talk a little bit about nutrition and an experiment that we did in collaboration with the West Plains beekeepers and was part of one of my graduate students' thesis projects. So we tested this dietary supplement, which was basically like an artificial nectar pollen mixture, and we mixed it with sugar water and used these inline feeders to expose some hives and feed them this artificial nectar pollen supplement, and then other ones we had control, which was just the sugar water. And so we were interested in seeing if this, of course, impacted the gut microbiome of the bees, but also if it improved the growth of the hive overall. And so we found that this diet treatment did not significantly affect the weight of the hive. And so we tracked these hives from April to October. And so the nectar treatment is the dotted line, and then the sugar control is the solid line. And so at each time point that we weighed the hives, there was not a significant difference. And then this is kind of the average hive weight, which was not different between nectar and the sugar control. So didn't affect the weight of the hive overall. And it also didn't impact the gut microbiome. This is for a subset. We had 16 total hives across 2 sites, and so this is showing one of the sites. And if you remember from this type of plot, the points are they're overlapping, they're not clustering, and when we do statistics, they're not significantly different. So this artificial diet didn't alter the microbiome, which is actually, probably a good thing in the end, because even though we only measured the weight of the hive, there could be other benefits that we didn't measure of this supplement that we tested. And so it's good that, it's not affecting the microbiome in a way that kind of disrupts it, even if it is beneficial later. So that's kind of the impact there. And we also think that the more external food sources, because we weren't restricting them, so they were still out foraging on whatever was available. And so, we think that that might be shaping the microbiome more than the in-hive feed as well. But one thing that we did find from this same study was really interesting. And so we looked at their microbiome that October when we were done with kind of that part of the study. And then we tracked them over the winter to see if they survived the winter. And what we found is that the hives that survived all had a very similar microbiome to each other. And this was different than the hives that died over the winter. And so it's that pre-winter microbiome that impacted their overwinter survival, which was super interesting. And we looked at this, and this pattern held even at the beginning of that season in April and all the other summertime points in between. their microbiome originally was associated with overwinter survival, even back in April before. And so this was super interesting. And as you can see, though, the sample sizes are pretty small. And so we are actually right now collaborating with Washington State University and Montana State University to scale this up and look at microbiome across, hundreds of hives instead of just a handful here with our preliminary study. So we're really excited about that and also trying to identify, okay, what exactly is it about these hives that survive, like which types of bacteria that we're driving these patterns. And so we will follow up on that as well. And then another thing that we found, so we kind of have this correlation with survival, overwinter survival in the microbiome. And we also found a correlation with the mite levels, the Varroa mites. And so again, these points are the microbiome of hives, of the bees in a hive, and they're color scaled to the mite load. And so again, we saw a significant relationship between the infection level and their microbiome. And we're still trying to think about why exactly this is. Remember, this is correlational. It's not saying that the microbiome caused, you know, higher or lower mite levels or vice versa. It's just a correlation. So now we're trying to figure out like what the mechanism is. And so I have some ideas about maybe the gut microbiome is producing these chemicals that we know it does, and that might be influencing the integrity of the exoskeleton in a way that maybe makes the Varroa mites not be able to attach as well. Or if there's some other factor, that we're not necessarily accounting for. But I would love to hear if you guys have ideas about that, that we could potentially explore what exactly is going on with this pattern. But pretty interesting there as well. So in conclusion, I hope that you've enjoyed hearing a little bit about our studies that have shown kind of the microbiome being influenced by the environment, and these factors like pesticides and antibiotics, pathogens like nosema, and parasites like the varroa mite. So we have a lot of, kind of correlational observational data that we are exploring. But the different bacterial strains, even within the same species, were affected differently. And also the microbiome, varies across the hives, and even when they're really close proximity. And so these different levels of variation are making it really difficult to evaluate like what is a healthy microbiome, and is that going to be consistent, you know, across the different environmental contexts. So a little bit complex, but we think that, continued research on the role of the gut microbiome and bee health and colony health has a lot of potential. And thinking about, developing probiotics. I know there's a couple commercially available probiotics out there, and I've actually done some experiments on those that I didn't talk about today, but I'd be happy to chat about too. Um, but I think there's room for kind of more probiotic products that are customized for particular problems. And so yeah, I'm really excited to hear what you guys think, and if you have any ideas. Um, and yeah, so with that, I would just like to thank you all for being here, and also thank my collaborators at different universities and nonprofits across the country, and also just really want to acknowledge the students, especially Shelby, Macy, and Bailey, who were master's students, and then lots of undergraduate students as well. And funding from the National Science Foundation to tackle this problem with, trying to help protect our bees and pollinator health, and really make sure that our agricultural industry um, is solid, with the bees. Also, I had a seed grant from Virginia Agricultural Council that really sparked my interest in this research. And then faculty research grants from Eastern Washington University. And just one more shout out to the West Plains Beekeepers Association that has been really helpful kind of from the moment I got here and getting set up with my research program at Eastern. And then even now we continue to benefit from their help with our campus bees. So thank you all for your time.
Thank you so much, Jenny. I love the colored bee suits with the Eastern logo within your students. That's a nice flair and better way to tell people apart out in the bee yard.
Yeah.
For anyone watching us live, please feel free to post some comments in the chat and we'll read them. We'll read them off to Jenny. Couple just questions that you led off with. I love the background of just kind of understanding that basic variability of like there's 10 basic uh, 10 basic bacterial species, but how they interact and their quantity and their subspecies or strains, that's amazing. So complex and complicated. I don't even think we even are scratching the surface. That's amazing. Um, I'd like to hear more on your thoughts, right? That comment about your pre-winter microbiome or those young winter bees or the biome of those bees to help increase survival. Um, that's fascinating. Do you have anything more to share on that or ideas of how to help beekeepers increase their fall microbiome? We have we have Western Washington and some areas that get that really dark knotweed honey, which we hear is not great for overwintering, the darker honeys. Wet climates and then the really cold, dry climates out in Eastern Washington.
Yeah, that's a good question. Yeah, I think that, and I imagine that the patterns with the microbiome leading up to the winter, will probably be different in different parts of even our state. And that would be really interesting if there's any beekeepers listening kind of on the western side of the state that would be interested in collaborating on this, because yeah, most of even expanding to with WSU and looking at some of those hives are still kind of Eastern Washington and exposed to that environment. And so that would be really interesting. And I would love to, yeah, have something on the other side of the state too to look at that. But yeah, I think it's gonna be important to identify what is it about those survival bees in those groups. And so trying to figure out, okay, what particular bacteria are there. So my inclination based on all of my research so far is that it's probably going to be the same types of bacteria that are present. It's just the abundance, the relative abundance, and that can drive those differences in those plots that we were seeing. And so, for example, some of the experiments that I've done that I didn't show today, the results with pesticide exposures and basically the lactobacillus group was significantly reduced in some of the pesticides that I tested. And then we kind of repopulated the lactobacillus with the probiotics, and it, increased back to levels similar to the control. And so, yes, so if we can identify which taxa are impacted, whether that's increased or decreased, then I would say, yeah, the target would be to kind of reestablish that community. So, yes, still lots more to do there.
Yeah, there's I've seen there's a handful of probiotics, commercial products out on the market. Still, the jury from the research community seems to be out on how beneficial these things are. But I know, right, general consensus is, well, it can't hurt. So it is really interesting to see how beneficial that might be moving forward. So looking
Mhm.
forward to ongoing research there. You made another comment around the mites and the different biome, just causing a different biome. Any hypotheses or preliminary research being done on mites and microbiome? I know there's been quite a bit about, you know, bee motility and the vitigelin and how mites actually are detrimental to the bees and that vector for disease, but anything more on the um, bacterial species or the microbiome, especially considered that the microbiome can help reduce some of these other diseases, as your cool little microscopic diagram showed.
Yes, yeah, that is a great question. I would say that first, trying to understand the directionality of the relationship. So, is it that it's the microbiome composition and thus the chemical signals that they're producing that is regulating whether or not varroa mites can attach and take hold? Or is it the presence of the mites and, all of the things that they're um, like providing, or I get that's not the best word, not providing, but giving to the bees, right? We know how viruses um and so forth are spread often through the mites, right? So is it something about the mites and their presence that then is changing the microbiome, and then we see the correlation? So I think step one will be to figure out the directionality of that relationship. And then the mechanism. Like I kind of mentioned a little bit, there's one study on not bees, but another insect species that has linked the microbiome and the chemicals that they produce to the integrity and the thickness of the cuticle and the exoskeleton of the insect. And then that changed whether or not the ectoparasites could take hold. And so I'm really curious to kind of build off of that hypothesis and that study in another insect to see if that holds with the bees. Um, and then yes, but yeah, it could definitely be so many different factors because the mites are also, feeding on the fat bodies of the bees, and maybe that is then kind of triggering some shift in the microbiome and like a dysbiosis. And then that's what we're seeing. So, with the data we have now, it's not clear what the direction is. And it could be bidirectional too, because with the human gut microbiome, we know that the relationship between the gut microbiome and our nervous system, our endocrine system, our immune system, there's a bi-directional relationship between those systems and the gut microbiome, whereas they can both influence each other. And so it could be that as well going on with the bees too. So yes.
Fantastic. And then you mentioned imidacloprid consumed by the lactobacillus. Anything else, right? I know there's a lot of pesticides, herbicides in the environment, things that we are adding as beekeepers to help our bees combat mites or other diseases and fungal infections. Anything else that has come out in the research of, again, proving more microbiome support can only help the bees be more successful against combating, our very toxic environment.
Yeah, definitely. Yeah, there's a couple studies that I cited today that have identified lactobacillus, but also other bee bacteria that can metabolize a lot of different chemicals. And so that in combination with figuring out like how they do it, I think it's going to be really important. Um, And I did another study the same one I was talking about with the pesticide exposures, so in the hive. And in that one, I tested talfluvalinate, and I didn't see an effect of talfluvalinate, which is one of the mite treatments on the microbiome. So that's good. That, not all of these treatments are kind of causing a shift in the microbiome that might impact the health of the bees. Um, but of course, we know about resistance, the mites evolving resistance to different treatments for the mites. So that's also another big concern too. So I talked a little bit about antibiotic resistance and how some of the bacteria are evolving resistance to tetracycline. And that another interesting study found that tetracycline resistance is way more prevalent in the United States, where people are using tetracycline, compared to Europe, where it's not. Um, I don't know if it's being used, but it's not supposed to be used in Europe. And so I think the overall amount of use in Europe is definitely less than the US. And they don't see tetracycline resistance in the bee gut bacteria in Europe at the levels that we're seeing it here in the US. Um, so that's definitely another thing. So just yeah, it's an arms race, right, between the Varroa mites and the treatments for Varroa, and it's an arms race between, using antibiotics, but then the bacteria are evolving resistance to that. And so I think one thing that I know a lot of beekeepers do is kind of rotate through their different types of mite treatments, right? And so we might need to think about that for tetracycline use and antibiotic use, right? As sort of like alternating different treatment methods as well. So, um, yeah, and like, discovering new antibiotics. That's another area that I'm interested in, is like antibiotic discovery and looking in new habitats, new environments for bacteria that are capable of producing antibiotics that haven't been used before and that our pathogens aren't resistant to yet, at least. So, um, yeah, so those are some of the things that come to mind with that question.
Awesome. It just seems like the more questions we ask, attempt to answer, the more questions it leads to, an ever evolving problem. So in the evolution. Um, just kind of, everyone asks about recommendations, wants to know the best products or the best way to do things. Do you have any recommendations from your research at this point, knowing that some of your research may be more lab-based, knowing that you're working on some field-based studies about treatment plans or even some probiotic plans, um, keeping in mind, right, the label is the law, commercial availability, all of that, um, that you would recommend to beekeepers at this point in your research?
Yeah, so the product that I tested that I was mentioning, and I didn't really talk too much about in my talk today, is I use the Super DFM direct feed. It's like Strong Microbials, I think is the company. That's the one that I used, and you basically mix it with powdered sugar and sprinkle it along the edges of the hive. And that's the one that I found, and it's mostly lactobacillus species in it. And that's what I found was actually able to successfully repopulate the lactobacillus population in the bee gut when it was decreased by chlorothalonil, which is a particular chemical pesticide. And so that was pretty interesting that it was, yeah, it was, the chemical knocked down the lactobacillus, and then I was able to repopulate it with that strong microbial super DFM product. And I haven't really tested that beyond that study. Um, and then and then I also have kind of like made my own probiotic concoctions and text tested them based on um, some data that we found from the field and kind of identified certain bacteria that we think are influenced by pesticides, and also showed that those were beneficial. But um, so that's kind of the only one that I could probably really say that I have very much experience with, and that it did show kind of an ability to repopulate the microbiome that was disrupted by the pesticides.
We did get one more follow-up question on on probiotics. It's understanding all the pre and probiotics are more human-based and are of no use to honeybees, and none survive into the gut, right? I think there's some studies that say that's probably true in humans as well.
Yeah.
Um, is it true? Is it not true in honeybees? You were smashing up bee guts and looking at them under microscopes. Can you, I mean, kind of expand on, I guess, the last bit of comments that you were making?
Yeah, definitely. Yeah, there is a lot of research on humans, that, the stomach environment that our supplements kind of go through first is completely different. It's super acidic and, just very different than the intestinal environment, which is very basic, right? Completely different pH. And so a lot of supplements don't make it through that acidic environment to the point of the intestines where they're needed. And so, yes, and that's so sort of like the delivery method, and is kind of a big area in human probiotic research, and sort of allowing the probiotic to persist through, the first part of the GI tract. With the bees, it's a little bit, it's not as dramatic, I guess, of a change in the GI tract. And um, and also based on kind of our results that show that the bacteria that are found in the probiotic product did make it to the hind gut specifically, where the lactobacillus is. It's in the hind gut. Um, I think that's less of a concern for bees because we're kind of able to see the species that we give them, all the way and find it in their hind gut. Um, and so I feel like that is less of a concern for the bees. But I think there's definitely, more research to be done, and like I said, like specific products that have different formulation of probiotics depending on the issue, whether it's like a pathogen issue and you want to repopulate the community, from a pathogen versus, pesticide exposure or some other chemical exposure. Um, so I think that's also there's potential there to kind of customize probiotics depending on the issue. But of course, we have to figure out which bacteria are impacted by those different factors. Um, yes. And another thing just to keep in mind with like pesticide exposures in general, and I know a lot of beekeepers are aware of this too, but thinking about their microbiomes as well, just coordinating and communicating and being notified by whatever, if there is some sort of application going on within the vicinity of your hive, and like just having that communication between applicators, pesticide chemical applicators, and the beekeepers is something I've been trying to really advocate for. Um, in order to, protect them, and doing it at, different time of the day, that doesn't affect the bees as much, or like different seasons, different, that the bees would then be less likely to be affected. So definitely always trying to advocate for better communication between applicators and beekeepers too.
Awesome. Thank you. One last question before we let you go, since we're past an hour. What advice would you give to a hobbyist beekeeper to maintain as healthy as a hive as possible, or a small group of hives?
Oh gosh. I'm definitely not a beekeeper myself, and I like to make sure everyone's aware of that. I forgot to mention that at the start, is that, I definitely value my collaborations with beekeepers. I've done some beekeeping, of course, over the years and have been trained by, very long time beekeepers. But yeah, as far as my general recommendation would be, I guess just like if there's an issue, um, trying to identify it as quickly as possible. And then also don't hesitate to reach out to researchers that might have the facilities and the resources to be able to help you identify the problem if you can't, kind of figure it out with what you have and your experience and your knowledge or your mentor's knowledge. Like I've said before, I really value my collaborations with beekeepers across the country now that I've been able to work with. And so, um, just know that, most of us are very excited and, really willing to work with people and actually think it really helps us too to be able to collect more samples. And so that's another thing that I would say is that don't hesitate to reach out to people that might be able to use molecular tools, sequencing, microscopes, that kind of thing to identify what the issue might be. So
I love it. Jenny, I just want to thank you on behalf of WASBA, Washington State Beekeepers, all the research you're doing on our microscopic organisms that keep our bees healthy, trying to move that forward. We look forward to you reaching out when you want some citizen science or citizen sample help throughout the state. WASBA and the local clubs have a great reach. So with that, thank you. Thank you for everyone that watches either live or in the future. We really appreciate your time.
great. Yeah, thanks again for having me. I really appreciate it. And I love bees and I love the motivation to be able to help conserve them the best we can. So thank you.
Thanks.